We're going to take a look at sensory pathways in this video. The word transduce means to convert. So sensory transduction is when we're going to convert the energy of the sensory stimulus into receptor potentials. And receptor potentials are the same thing as graded potentials. So we've actually already learned about receptor potentials because we already know about graded potentials. So we've just changed the name of graded potentials to receptor potentials. So in sensory neurons, instead of calling them graded potentials, we're just going to call them receptor potentials instead. So they're going to be exactly the same. They're going to be created by ion channels opening or closing. And when the ions move in or out of the sensory neuron, that's going to create a depolarization or hyperpolarization of the membrane potential. And these graded potentials can vary in size depending on the strength of the stimulus. And if threshold is reached, then we can generate an action potential in the sensory neuron. So let's just take a look at these receptor potentials. So we've got a sensory or afferent neuron here, and we've got different size stimuli here. So maybe we're going to be detecting touch, for example, with this sensory neuron. And with these sensory neurons, it's a little bit different. So their myelin sheath starts here, and where they actually are going to start their action potentials are actually at the first node of Ranvier. Okay, so that's a little bit different than the other neurons that we were looking at before that are going to start their action potentials at the axon hillock. So these actually in the sensory neurons are going to start at the first node of Ranvier, that first gap in their myelin sheath. So that's just a little bit different. Okay, so we've got our first stimulus that we're going to apply. It's a pretty small one. And if we put a probe inside of the dendrites here on our sensory neuron and we measure our receptor potentials or graded potentials here, they're not going to be very big. So we're going to be opening up some ion channels here. Maybe we're opening up some mechanically gated ion channels. They're responsive to touch, and that pressure will open up the gate. And then maybe we'll let some sodium ions in, and you can see that we depolarize the membrane potential, but it's not enough to reach threshold at that first node of Ranvier. And so we're not going to generate an action potential in the sensory neuron. And then if we apply a stronger stimulus, so maybe we're going to poke the skin harder, and now we're going to open up more mechanically gated ion channels here in the sensory neuron. We let more sodium ions in, and then notice that our receptor potential is going to be bigger. So notice that our receptor potentials, our graded potentials, can vary in size depending on the size of the stimulus. And when we reach our first node of Ranvier here, we are above threshold, and so we will generate some action potentials. So we do stay above threshold here with our stimulus, that we do generate several action potentials. And then our next stimulus, we're going to poke our skin even harder and open up even more mechanically gated ion channels, let even more sodium ions in, create an even bigger receptor potential or graded potential here. And then when we reach that first node of Ranvier, we will be above threshold, and we're going to generate even more action potentials because we're going to stay above threshold longer, and we're going to increase the frequency of the action potentials. Remember, that's frequency coding, and that's going to tell the brain that this is a much stronger stimulus. Okay, and so you can see if we measure the action potentials further down the sensory neuron, you can see here we've got more frequent action potentials with the bigger stimulus, and then you can see when we get to the end of the sensory neuron, these action potentials reaching the axon terminal will trigger the release of the neurotransmitter, and then we can talk to the next neuron in the pathway. Okay, the other thing that's really different with these sensory neurons is these sensory receptors will adapt, meaning that the receptor potentials, these graded potentials, will actually decrease in size over time with the presence of a constant stimulus. So this occurs with a constant stimulus. Okay, that's the key with the receptor adaptation. So if your graded potential or your receptor potential that you're producing, initially is big and then it gets smaller, if that stimulus is still being applied this whole time, then what's going to happen to the frequency of the action potentials in the afferent or sensory neurons? So if your receptor potentials decrease in size, those graded potentials decrease in size, you're no longer going to be reaching threshold, right? And so you won't be generating action potentials, and so that'll decrease the frequency of the action potentials, and so you won't be sending as many signals to the brain. And so how is this going to affect the perception of the stimulus? Well, you're not going to notice it as much. Okay, so we've got two different types of sensory receptors. We have sensory receptors that adapt to a constant stimulus very slowly, and these are called tonic receptors. So that would be really important to remember. That will probably be on the next exam. So make sure you know about tonic receptors. They adapt to a constant stimulus very slowly. And then these rapidly adapting sensory receptors are called phasic receptors. Okay, so let's just kind of give you some examples here. So a tonic receptor, one that adapts slowly, is always going to be sending your brain information about that sensory stimulus. So sensory receptors that adapt slowly would be like the proprioceptors, so like the muscle spindles in your muscles that detect muscle length, and the proprioceptors in your joints that detect, the angle of your joint. Those are always going to be sending signals to your brain, action potentials, because you always want to know the position of your limbs. Okay, that's pretty important information for your brain to know. So those are going to be examples of tonic receptors. Another example of a tonic receptor would be pain receptors. So unfortunately, if you're in pain, you're going to keep feeling it until whatever is causing the pain is resolved, because that's a pretty important stimulus. You don't want to be using a painful body part and doing more damage to it. So you're going to keep sending that pain signal, if things have not healed yet. Some examples of phasic receptors, ones that rapidly adapt to a constant stimulus. For example, this guy in the pool. When you jump into the pool and the water's cold, at first it feels freezing, but then after you've been in the water for a couple minutes, then it doesn't feel quite as bad, right? So those receptors that detect temperature on your skin, they're going to adapt pretty quickly to that change in the temperature from the shock of the cold water. And then they stop sending signals to the brain, and your brain's like, oh, the water's not so bad anymore. Okay. Another one would be your sense of smell. Those are phasic receptors. So like you walk into the anatomy room and at first it smells really bad. Okay, but then after a couple minutes, you don't notice it as much. Okay, because the sensory receptors picking up the sense of smell, they start to adapt and they're not sending as many signals to your brain, and then you don't notice the bad smell as much. Okay, so those would be examples of phasic receptors. All right, so let's just take a look at what's happening with receptor adaptation. So on the left, we've got some tonic sensory receptors. So this means they're going to be slowly adapting to a constant stimulus. So on the bottom in the pink, we've got a constant stimulus. So we're going to apply it, keep it on, and then here we're going to take it off. Okay. And then the green on the top is our receptor potential, our graded potentials. So in our sensory neuron, we apply the stimulus, we trigger our receptor potentials in our sensory neuron, and you can see we have a little bit of adaptation. Our receptor potential gets a little bit smaller, but we're still going to be producing a receptor potential in that sensory neuron. And we still could be sending some action potentials, but probably just not as many as what we did at first, okay, when the receptor potentials were bigger. And then when the stimulus is taken off, then we stop producing the receptor potentials. Okay, so these tonic receptors, they're going to adapt very slowly to a constant stimulus, and they're always going to be able to send some action potentials to the brain, letting you know what's going on with that constant stimulus that's still there. Okay, so like pain or your joint position, what are they doing? Okay. All right, here's an example on the right of a phasic receptor. So these will adapt rapidly. Some of these phasic receptors, when you apply the stimulus, they respond when the stimulus is first applied. So you get some receptor potentials in your sensory neuron. But then look how fast they adapt, and then they stop producing receptor potentials. So even though the stimulus is still there, they stop responding. Okay, and then they respond again when the stimulus stops. Okay, so a good example of this would be some of your touch receptors. So like when you sit down, the touch receptors in your rear end respond to that because you feel the pressure, okay, from the chair. So you would get these receptor potentials, and that would trigger some action potentials in those sensory neurons in your rear end, and you send some signals up to your brain to say, yep, we're sitting on the chair. And then you'd adapt very quickly, and then you wouldn't send any signals about your rear end sitting on the chair, okay, because you probably didn't notice that you were sitting on the chair until I said something. But then when you stand up, that stimulus has now changed, right? You no longer have that pressure, and so you're reacting to that change in that stimulus. And now you're going to send, you'll have another signal that, hey, things have changed. And so these phasic receptors are going to be responding to changes in the stimulus. So like when it's first applied and when it's taken off, but they're really not going to send signals when it's still there. Okay, so those are the phasic receptors. So they adapt rapidly. Okay, and then this is just another picture of receptor adaptation to a constant stimulus. So here's our sensory or afferent neuron, and we're going to apply our stimulus here in yellow this whole time. So we've got a constant stimulus, and let's take a look at our receptor potential measuring in our dendrites here. And you can see that when we first apply the stimulus, we get a bigger receptor potential, but then we slowly adapt and the receptor potential gets a little bit smaller. And then when the stimulus gets taken off, then of course we stop producing the receptor potentials. And if you look at the action potentials that we're producing here at that first node around VA where we start the action potentials, you can see that at first, where the receptor potentials are bigger, we get more frequent action potentials. And then where the receptor potentials are not quite as big, then we have less frequent action potentials. But we're still sending action potentials to the brain and letting the brain know that, hey, you know, our limb is in this position. You know, this is what your leg is doing. Okay. All right, and then over here on the right, we've got some phasic receptors that adapt rapidly to this constant stimulus. And some of these phasic receptors only react when you first apply the stimulus, and then they don't react when you take it off. So some of them will do that. So here it produces a receptor potential when you first apply the stimulus. So maybe you walk into the anatomy room and it stinks, and so your sensory neurons in your nose will produce some receptor potentials. We'll reach threshold at that first node around VA, send some action potentials to the brain and say it smells really bad in here, and then after that they adapt really quickly, stop producing the receptor potentials in the sensory neuron, and then you no longer send the action potentials to the brain, and then you get used to the smell. Okay. Let's take a look at these sensory pathways. Labeled lines are really important to know about. These will probably be on the next exam. So labeled lines are just specific neural pathways that transmit a particular sensory modality. And remember, a modality is just the type of stimulus. So whether it's light, sound, touch, smell, or whatever it is. So the brain has labeled certain pathways in the brain that, for example, let's say that you've got a neural pathway coming from the eyes. So anything that is traveling around from the nerves coming from the eyes and ending up in the visual cortex, in the occipital lobe in the back of the brain is going to label any of that information coming to that occipital lobe. It's going to interpret it as light, okay, because it just knows that, hey, we've got photoreceptors in the eye, they respond best to light, and so any of those nerve signals coming in that labeled line, traveling this pathway to the occipital lobe, the visual cortex back here, we're just going to interpret that as light. Okay, so the brain has just labeled that pathway, that's got to be light. Okay, but what happens when you push on your eyeball? Okay, and so if you push on your eyeball, you see stars, don't you? You see little flashes of light, because those photoreceptors can also respond to pressure, but because the brain has labeled that pathway coming from the eye to be interpreted as light, any signal coming through that pathway, even though it was actually pressure that stimulated the photoreceptors, the brain is going to interpret that as light. So that's why when you push on your eyeballs, you see stars. You see those little flashes of light. So that's pretty cool, isn't it? Okay, so that has to do with these labeled lines. Okay, seeing stars is from the pressure on your eyeballs. Those flashes of light, that's because of these labeled lines. Okay, so organization of these sensory pathways, we have these sensory units. That is just talking about these sensory or afferent neuron with all of the receptors that it has. So there might just be some free nerve endings here, just some dendrites, or maybe there's some specialized receptor cells before the sensory or afferent neurons. So this whole thing would be the sensory unit. And then we also talk about a receptive field, and in this neuron on the far right, you can see this purple area. That is all the receptive field for this particular sensory neuron. And this receptive field is just the area over which an adequate stimulus can produce a response in the efferent neuron. So maybe this sensory neuron is going to detect touch. And so if you were to take a needle and poke the skin anywhere in this receptive field, then this sensory neuron would be able to pick that up. So remember, in our sensory pathways, it takes 3 neurons to get from the periphery up to the cerebral cortex. And so we've actually named the 3 neurons. So this would be important to know for the exam. The first order neuron, that's going to go from the periphery up to the spinal cord or the brainstem. So this sensory neuron here, this will be the first order neuron. And then the second order neuron is an interneuron that travels from the spinal cord or brainstem up to the thalamus, and then synapses in the thalamus. And remember, the thalamus, that's your filter, and it's going to decide if that sensory information is important and send it through, or if it decides it's not important, it'll just stop the information right there and not let it through. And then the third order neuron is going to go from the thalamus up to the cerebral cortex. And then you'll be aware of whatever the sensory stimulus is. So here's just an example of the gustatory pathway. That's the fancy medical term for taste. So we've got taste buds in the tongue, and those are the specialized receptor cells for the sensory neurons for taste. And so the first order neuron will start in the periphery here in the tongue, and then travel up and synapse with the second order neuron in the brainstem. And then the second order neuron will travel up to the thalamus, synapse there with the third order neuron, and then the third order neuron will then synapse in the cerebral cortex, where there you'll be aware of whatever it is you are tasting or eating.